Hello and welcome to the Beyond Biotech podcast number 108. I'm Jim Cornwall and this is the weekly podcast from Labiotech. This week we have an in-depth conversation about combination vaccines, from what they are to what they can be used for via COVID and bird flu, and what the future holds for combination vaccines. And who better to walk us through the subject than Cesar Sanz Rodriguez, Vice President, Europe and Switzerland, Medical Affairs at Moderna. So I guess the first question then is if you could tell me a little bit about the technology that allows for a combination vaccine and how that was developed.
Yeah, so as you know, combination vaccines do have a huge potential for impacting public health and global health outcomes. And for that very same reason, they've been really top of mind for clinical developers over time. So actually, the interest in developing combination vaccines goes back to the 1940s, when the first combination vaccine was developed. I'm talking about the DTP vaccine, which targets diphtheria, tetanus, and pertussis, and that was essentially a combination of toxoids. But then over time, with advancements in immunology, molecular biology, and biotechnology, there's been a number of advancements that have enabled further developments. So for instance, the use of adjuvants to enhance immunogenicity, the application of DNA technology, recombinant DNA technology in the 70s and 80s. And the hepatitis vaccines are good examples of that. Or also the use of subunit and conjugate vaccines are good examples of how that progress has been made. In recent years, we have seen more sophisticated technology advancements. So I would say there's primarily two. One is the use of virus-like particles. These are essentially particles that mimic the structure of a virus, but do not contain any genetic material from the virus. So that makes them very safe. And the other one is connected to advances in genetic engineering and synthetic biology, which allows you to design antigens as you wish. And then also essentially the advancements in nanotechnology, which have helped with the delivery of such antigens. So this all comes together actually in the mRNA technology, which was proven to be very powerful during the COVID-19 pandemic, as you all know. In fact, the mRNA technology is also very well suited for the development of combination vaccines, because you can actually design multiple antigens, and also the scalability and the flexibility of the platform allows for very rapid production times. So that's the reason why it's been really one of the areas of attention recently.
What are the main advantages of vaccines that target multiple respiratory viruses at the same time?
There are actually several, I would say, significant advantages, and these benefits extend both to individuals, who benefit in terms of health outcomes, but then also to the societal perspective of public health and healthcare systems altogether. So to me, the first one I would like to highlight is the opportunity for improved vaccination coverage and compliance. And this really comes with the simplicity that combination vaccines offer. Simplicity because, number one, there are fewer injections, and that really helps some of us who are a bit scared of needles become more inclined to accept vaccination. Also, vaccination calendars are getting busy. There are many different vaccines that, we need to protect against. And obviously, having the opportunity to combine different pathogens in one vaccine, that simplifies also the compliance with those vaccination calendars. Altogether, this translates into enhanced public health outcomes. So we have broader protection in the population. We increase the overall immunity in the population. And also by preventing diseases, we reduce the burden of disease associated with those infections. And so, for instance, if you take the example of respiratory infections, we are now thinking about, what's going to happen in the fall, for instance, with the influenza and the COVID-19 vaccine campaigns. Prevention of such respiratory infections will lead to fewer hospitalizations, medical visits, and complications. And then on top of that, there's also, I would say, economic and logistical benefits. So first of all, in terms of cost effectiveness, the prevention of multiple diseases through the use of a single vaccine can definitely help reduce the overall healthcare cost associated with treating those infections. And also it allows for a more efficient use of resources. So, resources that can be spared by using combination vaccines can then be redeployed for other purposes and to address other areas of unmet medical need. And this is particularly true in context of low resource access, which is the case in some countries across the globe. So I think for all these reasons, we have seen vaccination with combination vaccines become quite a standard in pediatric, for instance, in the pediatric segment. And there is also, I think, a big opportunity to have some of these benefits also in other patient groups at high risk for infections, such as the elderly, people with immunocompromised immune systems, or essentially people who have, one or more underlying conditions. They all may benefit from the use of combination vaccines.
You mentioning the cost, there's probably also a good case to be made for the fact that people don't have to travel 3 times to get 3 injections. They only have to travel once. So that's probably a savings environmentally as well.
Absolutely. But I think, from my point of view, that cost is there, of course, but I think it's more about, again, making vaccination more friendly to those of us who may benefit from it.
In terms of the efficacy, how does the immune system tackle dealing with multiple pathogens at once?
So the risk of infection produced by different pathogens at the same time is actually something very real we deal with as human beings. So I'm sure many of our listeners have actually read recently in the newspapers about the concept of the triploidemic, which is essentially the big surge of infections we have seen associated with the influenza virus, the SARS-CoV-2 virus, which is the virus causing COVID-19, and then also the respiratory syncytial virus. And it's not only that they produce many infections, they actually sometimes happen at the same time, which makes the situation more complex for those affected. So the good news is that our immune system is actually well equipped to deal with that. And that is because of, I would say, 3 elements, 3 key elements of our immune system. So first of all, we have a diverse repertoire of B cells. These are the white blood cells that produce antibodies. We also have, T cells, T lymphocytes, the so-called helper T cells or CD4+, which actually help also in the development of that immune response. And then within the lymphoid organs, there are some very specialized structures called germinal centers, which also help in that process. So essentially what happens is that, when the immune system is exposed to antigens present in those pathogens that are co-infecting, different B cells will get activated. So essentially each activated B cell clone will produce a specific type of antibody, but altogether we have different clones that are being activated. So we are able to really produce antibodies against all those pathogens. And this is actually the same principle that we take advantage of when we think of designing combination vaccines. Essentially in these vaccines, we bring together different antigens, and in doing so, we will essentially trigger an antibody response against all those antigens by different B cell clones that will be activated.
Are there any differences in efficacy or safety between giving multiple vaccines separately as opposed to altogether?
No, I'm really happy you're asking that question, because that's actually a question oftentimes people have when they think of taking a combination vaccine as opposed to the individual counterparts. Generally speaking, combination vaccines are designed to be as effective and as safe as their individual counterparts. And this is very clearly outlined in guidance that different regulatory agencies have produced. This is the case, for instance, for the FDA in the USA and the European Medicines Agency in the European Union. In fact, just to illustrate this point, the European Medicines Agency, the EMA, has actually developed a guidance that includes very clearly detailed instructions as to how a vaccine should be assessed, both from an efficacy and a safety point of view. So when it comes to efficacy, the efficacy of combination vaccines is actually assessed based on immunogenicity studies, how strong of an immune response they are able to produce. Clinical efficacy trials may also be required. And then also, very importantly, companies are also required to demonstrate that the efficacy of the vaccine is actually consistent across the multiple production batches, so that there is essentially a reliable vaccine to be used in a broader scale. And then the safety of the combination vaccines is also extensively evaluated. And again, this is based on preclinical studies, including toxicology studies. Again, clinical safety trials may be important. Once the vaccine is in use, there's also a very broad program of post-marketing surveillance. So essentially all the pharmacovigilance mechanisms are in place to understand if there's any new safety concern. And also oftentimes vaccine manufacturers are required to put in place the so-called risk management plans. And these are important to not only identify, but also to mitigate potential risks associated with the use of combination vaccines. So I think the message I would like to close with is really that clinicians and the population overall should all be reassured that when they see a combination vaccine, even if it's a combination vaccine that has been developed with the newer technologies I was talking about before, these vaccines are actually very rigorously assessed, and they meet all stringent and also very comprehensive criteria for efficacy and safety.
And I assume in terms of boosters, we've had the COVID where we needed to get one every, I don't know, it changed over time, but would boosters still be required? And how would you manage the fact that maybe some of these have different lengths of or periods of time between injections?
So boosters are sometimes required for combination vaccines, but just as they are as well for some of the single antigen vaccines. So in fact, the need for a booster is dependent on a number of factors, and these have to do with the nature of the pathogens involved, the type of vaccine platform that we are using, the immune response elicited by the initial doses, the so-called primary series of the vaccine, and then also the duration of the protection conferred. Again, just to give you some examples, the need for boosters in combination vaccines is not new. It's not a new topic. So before I was referring to the combination vaccine that targets diphtheria, tetanus, and pertussis. So, for those listeners who may have young children, they may be familiar with this vaccine because actually their kids had to take several doses of it when they were during infancy and early childhood. But actually this vaccine requires a booster during adolescence. And even in adult age, for instance, pregnant women will need to take a booster to protect the new infants from pertussis. Or another example could be the hepatitis A and B combination vaccines. Again, this vaccine in particular, for instance, requires a booster when given to patients who have an immunocompromised condition. So again, it's something that is not new. The point I would like to make here is that actually newer technologies such as the mRNA technology, as you were bringing up in the question, could actually be very helpful also in this context, because all the flexibility, all the adaptability that they bring, that can actually be applied to the development of boosters. And to the example that you were using, this has been shown to be very much the case with the adaptation of the vaccine, the COVID-19 vaccine, to the different variants that have been coming up. So I think in any case, what's important to keep in mind is that public health authorities and clinical developers are the ones who are continuously assessing the potential need for a booster, and they do that based on scientific evidence as well as on epidemiological data. So no one should take any decision, I would say, individually for a booster vaccine. Again, that is really the call of public health authorities.
Are there many companies that are working on this kind of technology?
I would say, pretty much any company who plays in the field of vaccines is actually working on developing one or more, combination vaccines. I would like perhaps to bring it back to mRNA technology because, again, as I said before, this is one technology that really has a lot of potential and can be very instrumental in the development of combination vaccines. So at Moderna, for instance, we do have actually a number of projects underway looking at combination vaccines. The most advanced one is actually looking at a combination of influenza and SARS-CoV-2, but we actually have different programs looking at different combinations of other respiratory viruses, including the two that I just mentioned, but also, for instance, respiratory syncytial virus, also known as RSV, and human metapneumovirus, also known as HMPV. Our colleagues from BioNTech are also working on a combination vaccine targeting influenza and SARS-CoV-2. I'm sure we're going to see lots of good news in this space, coming from different companies in the years to come.
In the news just recently, we've heard about the declaration just this week of mpox as being a major issue. And before that, we were talking about potentially bird flu making its way into the general population. How easy is it to add a new vaccine into this kind of technology?
It's not easy, but it doesn't mean it cannot be done, and it doesn't mean it's not a priority for all us working in the development of new vaccines. So, I'd like to perhaps spend a bit of time talking about avian flu, because I really think, there is a major concern, I would say, around that one in particular. As you know, in the news, it's been quite present in the last few months, particularly because of some cases, where the virus was able to jump from mammals to humans. In fact, avian flu viruses have been recognized as a threat to human health for decades. Back since the late 1990s, early 2000s, when there were some H5N1 outbreaks, that concern has been there. And in fact, that is the reason why many companies have actually been putting a lot of work. Not only companies, also academic research groups have been putting a lot of attention to this particular virus, and specifically a couple of strains which are highly pathogenic, such as 5H1, which I already mentioned, but then also H7N9, which is an ongoing focus of research. Fortunately, there have already been a few inactivated adjuvanted vaccines developed against H5N1, which have allowed for the basis of some emergency stockpiles. But the reality is that we need to be better prepared for a potential pandemic, and that is a real threat. And in that sense, the different technologies that I was talking about are also being applied to such research. Now, coming to mRNA technology, there is a sentiment that it can actually be very instrumental, and I would say there's two reasons why this is the case. Number one is the virus variability. Avian flu viruses actually mutate very rapidly. That's one of the characteristics they have. So having a technology that gives you the flexibility to essentially adapt the sequence that you need to target, and that's very important because it allows you to keep up with the pace of mutation of that virus. And again, we've seen that proven to be the case in the pandemic, in the COVID-19 pandemic. The other aspect is that because mRNA technology does not depend on biological systems for production, it can be very easily scalable. And this is also a very important feature, particularly when you think of a pandemic potential risk. So in fact, we do have a program right now. It's in phase 2 clinical testing, a program to evaluate a vaccine candidate against the prevention of pandemic influenza. We are really encouraged, and in fact, just as a testimony to how much attention there is to, the potential of this technology for this particular infection, this program is actually receiving partial funding by BARDA. So, I think that's just, an example of how important the potential can be. Another aspect that could come into the discussion is once we have such vaccines available against avian flu, how do we manage them? So you were talking about the vaccination calendar. How do we fit them in? This is actually a very complex discussion. There's many or decision. There's actually many factors that play. I don't think there's probably at this point in time a need to think that they should be part of the routine calendar vaccination, but perhaps a more targeted approach. Focus more on protecting the higher risk groups, and also enhancing pandemic preparedness and surveillance. That may be more practical and cost effective, and in that sense it may be perhaps a better approach right now to think of how we would manage the threat of avian flu.
How practical is it to add vaccines to a combination vaccine? I mean, is there a limit to the number that you could include? I know we were talking about 2 or 3, but could you do 4 or 5 in the same vaccination?
It's always difficult to talk about limits when you discuss technology and science, because the reality is that with time and advancements, typically the limits can be pushed further. So when it comes to combination vaccines, we've been exploring up to 3 different pathogens in one vaccine. But for instance, if you take the example of another vaccine, which I didn't talk about, but which we are developing, which is we are developing a vaccine against cytomegalovirus, which is a virus that can actually produce congenital malformations if the mom is infected, while she's pregnant. In that particular one, we've been able to actually combine in one vaccine 6 different antigens. Again, I'm pretty sure that over time we'll see more advancements also in this space, and while probably now 2-3 seem to be the more reasonable number to go with, I wouldn't be surprised if we saw more. And in fact, there are some examples also in the market, in the pediatric vaccination scheme where you actually see more than several vaccines combined.
Obviously, testing is massively important with all of this, but how easy is it to change or update a combination vaccine, for example, when, as you mentioned, with some of the fast mutating viruses? I know we had all of the different COVID variants.
If we take the example of the mRNA vaccines, this can actually be a very straightforward process. And again, we've seen it already in play a number of times for the COVID-19 vaccines. More recently, the adaptation of these vaccines to the JN1 lineages as recommended by regulatory agencies. Essentially, once you know the genetic sequence of the new variants, you just need to synthesize the corresponding mRNA sequence. This is something that is supported by great technological advancements also in the IT space, but essentially this process can just take place within a few weeks. then you may need to do some testing. It could be only preclinical. There could be a need for, also clinical trials, which could take also perhaps a few months. And this would depend obviously on regulatory requirements. But I think the very important element here is that even though you need to adapt the sequence as I was describing, the production process essentially does not really get impacted by the need of major modifications. So altogether, this really allows that, within a matter of a few months, you can actually update a vaccine. And you could do it for, a single antigen, or you could do it for different antigens, depending on what the need is. And then obviously the synthetic nature of the mRNA production allows also for a very rapid scale up based on the global needs. So again, I think this seems to be, at least for the mRNA technology, this could be a very straightforward process.
What do you see as the future for combination vaccines? It seems like it's a very, not only an exciting area, but also a very fast developing one.
Yeah, I think that the future of combination vaccines is definitely looking very promising. Again, I think all these advances that I was discussing before in immunology, in biotechnology, also how public health strategies are being shaped to incorporate these vaccines more effectively. I think this all offers a lot of, I would say, potential for significant impact. But let me perhaps now talk a bit more about one aspect I didn't touch so much about before. We've been talking about the technological advancements, but I think one element which is also of critical importance is how quickly this can happen. Classically, when you think of development of combination vaccines, that typically spans 10-15 years, from initial research to regulatory approval and full deployment of the vaccine. Coming back to the mRNA technology, I think, for the reasons I've been mentioning, I think that technology has the potential to really deliver on much shorter timelines. It's about, the flexibility that it offers, the adaptability, the possibility to rapidly design and upscale production capabilities. So, I think in that sense, we're going to see not only newer vaccines, more effective vaccines, hopefully as well, but also faster development and I think that's actually something that really makes this field particularly exciting and makes me feel very passionate about it. What I think is important to note is that we will need to continue to invest in research, infrastructure, and also in international collaboration will be very important to make sure that, recommendations and implementation is aligned across different countries to make it more effective.
In terms of rollout of some of the things that you and other companies are working on, what kind of timelines are we looking at for some of those?
If I look into our pipeline, which is what I know best, and I would like to perhaps focus on, just to be fair, and not to make comments on other companies' pipelines, but I do think we're going to be able to see some really good additions to the vaccine armamentarium within a couple of years in many cases, and then we'll see more to come. So again, I think it's going to get very, very exciting and interesting. And I do think the way we think of combination vaccines, which historically has been very much confined to the pediatric space, is going to change, and we're going to be able to offer such solutions to other population segments also at risk of infections.
Is there anything else that you wanted to highlight that we didn't talk about?
To me, what's very important is to leave this message that again, we're living very exciting times when it comes to the development of combination vaccines. And I do think the broader availability of such vaccines, the newer technologies that are becoming available to all of us, I think that's really going to help us improve the perspectives and to improve essentially the global health outcomes of vaccination, which is one of the most cost-effective medical interventions you can think of when it comes to public health. So again, I think we're living really exciting times, and I'm very much looking forward to seeing those innovations come into fruition.
A fascinating subject, and definitely one that's risen in prominence since COVID. And of course, there are plenty of other potential pandemics lurking out there, so vaccines will always be relevant to stay ahead of the curve. Don't forget to check out the latest news and articles over at Labiotech.eu, and I hope wherever in the world you are, you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Beyond Biotech.